Welcome to the AQuA Sample Analysis training video. In this video we'll show you how to measure phosphate concentrations using the hanometer, prepare and incubate your E. coli samples, safely handle and dispose of materials, upload your results for phosphate and E. coli to the AQuA Data Collection app, and ship the remaining samples to the University of York for analysis. On the day you collect your field samples, you'll also need to do the phosphate analysis and prepare your E. coli samples. Then, 24 hours later, you'll return to the Petrifilm to count any E. coli colony-forming units, or CFU, that grew. Before beginning, read the risk assessment and use the personal protective equipment supplied with your kit. When you upload your results, you will be asked to confirm you read the risk assessment. Before beginning the phosphate and E. coli analysis, prepare a clean workspace away from food preparation areas, children and pets. Clean the work surface using the antibacterial wipes provided in your kit and gather all of the materials you will need before starting. Throughout the analysis, wear the disposable gloves, eye protection and a face mask when doing the phosphate test. The HANA phosphate test uses a very small amount of reagent supplied in a sealed single-use sachet. Avoid inhaling the powder and open the sachet carefully away from your face. If any reagent comes into contact with your eyes, rinse thoroughly with water and seek medical advice. If you get it on your skin, wash the area thoroughly with water. By following these simple precautions, you can safely carry out the AQuA home analysis while generating high-quality environmental data. First, you will measure phosphate concentrations in the water you collected using the hanometer. This measures phosphates using a colour reaction inside a small glass bottle called a cuvette. Phosphate is an important nutrient in aquatic ecosystems, but high concentrations can contribute to algal growth and reduced water quality. To get started, locate the filtered phosphate sample collected in the field. Make sure the water is at room temperature before you begin the test. You can do this by leaving it out on the table for an hour or so, or holding the vial in your hands for a few minutes. Carefully pour the sample into the cuvette up to the 10 milliliter fill line. Use the lint-free cloth to remove fingerprints or water droplets from the outside of the cuvette, as these can affect the reading. Turn on the hanometer by pressing the button and insert the cuvette. The display should read C1. Press the button again to zero the instrument. Wait until the display changes to C2. Remove the cuvette and carefully open a phosphate reagent sachet. Add the full contents to the cuvette and securely replace the cap. Gently swirl the sample until the reagent dissolves completely. This may take a few minutes. Avoid shaking vigorously, as bubbles may interfere with the measurement. Insert the cuvette back into the hanometer. Press and hold the button until the timer appears. The instrument will now begin the 3-minute analysis. At the end of the countdown, the phosphate concentration will be displayed on the screen. Write this value onto the result sheet provided and keep it with your E. coli samples so you can enter all results together into the AQuA EpiCollect survey the following day. Next, you will prepare the E. coli test. The E. coli test allows us to estimate the amount of E. coli bacteria present in the water sample. E. coli is an indicator of fecal contamination in rivers and lakes, and is important to study because it helps us understand where pollution may be affecting water quality, wildlife, and how people safely use and enjoy these environments. Be sure to wear gloves and eye protection when carrying out the test. These are needed because bacteria grown on the Petri film could potentially cause gastrointestinal illness if mishandled. Make sure you don't touch your face even with your gloves on. Then assemble the incubation box. Remove the hand warmer from its packaging and gently shake it for about 15 seconds to activate it. It will not become hot immediately. Place the hand warmer inside the incubation box. Next, label the Petri film using the site labels provided. Be sure each Petri film matches the correct sampling location. Using the sterile syringe provided in your kit, draw up 1 milliliter of water from the E. coli sample vial. Carefully lift the Petri film cover and gently dispense the samples into the center of the film. Lower the cover slowly and gently tap the film to spread the sample evenly. Avoid pressing too hard or creating bubbles. Once the water sample has been added, keep the protective cover over the Petri film at all times and do not reopen it. Place the Petri film inside the incubation box on top of the hand warmer and loosely close the box, leaving approximately 1 centimeter for air to enter the box. You can stack multiple Petri films on top of each other. The sample should now be left undisturbed in the incubation box in a warm location for 24 hours while the bacteria grow. Make sure this is somewhere where people and pets cannot disturb it. Wash your hands thoroughly after handling the Petri film and disinfect your work surface both before and after use. 24 hours later, you can return to count and record the results. Remove the Petri film from the incubation box and place it in a plastic Ziploc bag. Then wash your hands or clean them with one of the antimicrobial wipes in your kit. Examine any blue dots that have formed. These are colony-forming units, or CFUs, of E. coli bacteria. It may help to hold the Petri film in the bag up to a light or window while counting. You may also see very small red dots on the Petri film. These are not E. coli, but other types of bacteria. In the AQuA project, we are not counting the small red dots. Some samples may contain only a few colonies, while others may contain many. Carefully count the number of blue CFUs on your Petri film. If there are a large number of CFUs, you may find it easier to count half of the Petri film using the grid and multiply the result by 2. You can also take a photograph and count the colonies on a larger computer screen or on your phone if that's easier. Each blue colony grew from bacteria that were present in the 1 milliliter of river water. However, water quality results for E. coli are commonly reported in the standard units of CFU per 100 milliliters. Using a standard unit like this makes it easier to compare results between different rivers, sampling sites and water quality guidelines. Because only 1 milliliter of water was added to the Petri film, you will need to multiply the number of blue colonies you counted by 100 to calculate the final results in CFU per 100 milliliters. For example, if you counted 12 blue colonies, the final result would be 1200 CFU per 100 milliliters. You're now ready to record your data in the AQuA Data Collection app. Follow the questions on the app to provide as much information as possible. Using the results sheet you completed during the phosphate analysis, enter your phosphate result, your E. coli result, remembering to multiply the colonies you counted by 100, and a photograph of the petrifilm. To take this photo via the EpiCollect app, hold the petrifilm in its bag up against a bright light source or window and take a clear image. The photograph will be run through image analysis software, so we will have a second count of the E. coli colonies. After recording your results, dispose of the petrifilm safely. Place the used petrifilms already in the sealed plastic bag into the paper cup provided in your kit. Carefully fill the cup with boiling water from a kettle and allow the materials to cool completely before disposing of the petrifilm in household waste. Finally, disinfect the workspace with one of the antibacterial wipes and wash your hands thoroughly. Once you've completed the tests and recorded your results, the next step is sending the remaining samples to the University of York for analysis. These are the samples for nutrients, metals and pharmaceuticals and personal care products. To prepare the shipment, remove these samples from the freezer along with the frozen ice packs. Place the samples and ice packs back into the insulated box that the toolkit originally arrived in. This box is designed to help keep the samples cool during transport. If needed, you may wish to add some newspaper or other padding round the sample bags to help prevent the bottles from moving around during shipment. Before sealing the box, double check that all sample bottles are labelled correctly. The lids are tightly closed and all frozen samples and ice packs have been included. Attach the prepaid Royal Mail shipping label provided with your kit to the outside of the box. The package can then be dropped off at any post office. To help prevent delays in transit, samples should only be posted on a Monday or Tuesday. Please avoid shipping samples later in the week as they may become delayed over the weekend before arriving at the university. If the samples warm up over a period of time, they may begin to degrade. If you need to wait until a Monday or Tuesday to ship the samples, just keep them in the freezer until you're ready. Once you've shipped the samples, please email to let us know that the package is on the way. Thank you again for taking part in the AQuA project. By collecting and analysing these samples, you are helping to build a better understanding of water quality and pollution in rivers and lakes across Yorkshire. One of the unique strengths of the AQuA approach is that community groups across Yorkshire are all following the same sampling methods, using the same equipment and collecting the same types of data. This means the information collected by different groups can be compared much more reliably across rivers, catchments and regions. By working together in this coordinated way, the data becomes far more powerful than individual samples collected in isolation, and help us build a much bigger picture of river health across Yorkshire. Working alongside our partners, including the Rivers Trust and Environment Agency, your results will support future environmental monitoring, research and community-led action to help protect freshwater ecosystems. Once the laboratory analysis has been completed, results from the project will begin to appear online through the AQuA platform, where everyone will be able to explore and compare findings from across the region. Every sample collected and analysed contributes to a bigger picture of river health. Thank you for being part of it.